Sanitary measures. Before entering the greenhouse, disinfect the soles of your shoes and any portable equipment you want to bring in on the phytosanitary mat with lime, located in the anteroom of the greenhouse. First, put on a coat. Afterwards, wash your hands with abundant soap and water. Then, rinse them well. After that, dry them diligently with paper towel. Last, disinfect your hands with 70% alcohol. Finally, rinse them again using an iodoform disinfectant such as Vanodin, a wide-spectrum detergent with immediate action against microorganisms. This procedure is an indispensable requirement before starting and after finalizing the working day in the greenhouse to prevent genetic material contamination. Greenhouse management. Planting progenitors. The clones selected as progenitors are provisionally grown in a greenhouse until their transfer to the definitive greenhouse where the crossing block will be established. We recommend the use of 8 to 10 tubers per progenitor. Individually, plant tubers that have 3 to 5 centimeters in diameter and good-sized sprouts in 4-inch pots that contain a substrate composed of soil and moss in a proportion of 2 to 1. Then, cover the tubers with the same substrate. After that, distribute them in an orderly manner on tables, where you will leave the plants to grow for a period of approximately 15 days before transferring them to the crossing greenhouse. Conditions and conditioning the crossing greenhouse. The greenhouse where the crossing block is installed does not require temperature and humidity control because the crosses take place during the most favorable time of the year for potato growth in the central highlands of Peru from November to March and from June to September. However, these factors can be monitored by means of sensors available in the market. In the cases where you have progenitors that require a long photo period in order to flower, the greenhouse must have a system of artificial lighting to extend daylight for up to 16 hours. Therefore, we recommend the combination of 20-watt white light bulbs and 100-watt yellow light bulbs, which provide the spectral width required to maintain the photosynthetic process to induce flowering. As part of the lighting system, install a timer that automatically controls turning on and off the lights at a given schedule. Crossing block installation. First, place the bamboo canes that will serve to support the plants during their growth. Press them in the ground at intervals of 30 centimeters. Then, fill the 10-inch pots 3/4 full with substrate composed of soil and moss in a proportion of 2 to 1. Afterwards, add 3 to 4 grams of fertilizer, prepared with a dose of 180 units of nitrogen, 160 of phosphorus, and 160 of potassium. For example, if you require 20 kilograms of fertilizer, use 7.8 kilograms of ammonium nitrate, 7 kilograms of diammonium phosphate, and 5.5 kilograms of potassium chloride. After that, add more substrate, manually compressing it with help of a wooden mallet. Repeat this operation several times until the volume of the pot is full. Last, lay down the substrate with the base of another pot. Finally, distribute the pots with the substrate firmly established alongside each bamboo cane. When this procedure is completed, the greenhouse is ready to have the plants that will be used in the crossing block installation. These plants must have reached an approximate height of 15 centimeters in the time they were kept in the greenhouse where they were previously planted. Before transferring the plants as part of a routine screening, you must test them for the potato virus T and the potato spindle tuber virus, which are transmitted to the seeds from infected plants. Then, maintaining aseptic conditions, collect 1 or 2 folios of the apical part of the plant in a small polyethylene bag, previously identified with the code of the clone and the number assigned to the plant. Then, send the samples to a laboratory that offers this service. According to the results, if there are infected plants, you have to eliminate them together with those in their surroundings, including those for which the results were negative. We recommend the repetition of these tests for every plant, even those with negative results. Unfortunately, if you find more infected plants, you will have to eliminate the whole crossing block. To install a plant, first, remove it from the pot, turning the pot over and tapping the base to receive the plant along with its substrate. Then, place the plant on one of the pots containing the compacted soil. After that, attach the plant to a bamboo cane with 1 or 2 pieces of thick thread. 10 days afterwards, remove the substrate covering the mother tuber with running water, exposing the roots. This procedure will also expose the stolons, which you should remove as they develop, carefully cutting them with a scalpel, previously disinfected in calcium hypochlorite solution. This procedure will induce the plant to remain in the flowering stage as long as possible and will not change its development from the flowering stage to tuberization and senescence. Finally, keep the plant in an upright position during its growth, tying it to the bamboo cane with additional thick thread. This will facilitate the crossing work, avoiding the plant's damage. Good soil texture, proper fertilization, and appropriate plant handling will ensure successful growth, profuse flowering, and finally, obtaining berries. Crosses. When the flowering season arrives, collect the flowers of the plants that you will use as male progenitors. For this purpose, collect completely open flowers with the anthers near dehiscence or completely dehiscent. You can recognize this by the light brown color of the apical pore. Afterwards, separate the flowers from the plant by the peduncle and place them in a piece of parchment paper or craft paper bag, correctly identified with the progenitor's number or code. Then, take these flower samples to the laboratory. Disinfect your hands with an alcohol swab before extracting the anthers one by one with the help of tweezers. You can also carry out this same procedure by hand. In order to do this, first, remove the pistil and place the cone of anthers on a piece of parchment paper. Then, fold it to one side to keep the anthers inside and fold the ends to close it and prevent the pollen from falling during handling. After that, put the envelopes that contain the anthers to dry for 24 hours on a table with 100 watt incandescent bulb installed, keeping a distance of 48 centimeters between the light bulbs and the table. The aim is to maintain an optimum temperature of 25 degrees Celsius. 24 hours later, extract the pollen. For this purpose, first, disinfect your hands and the equipment you will use with alcohol. Then, shake the anthers vigorously, holding a vibrator against the external part of the envelope to help release the pollen. After that, open the envelope carefully. Remove the anthers, gather the pollen, and distribute it into one or more gelatin capsules, properly identified using a fine point indelible marker. Following that, close the capsule tightly and tap it so that the pollen drops to its bottom. Last, place the capsule in a wooden container, specially made for storing several capsules in an upright position. Finally, disinfect your hands, vibrator, and table once again before extracting the pollen from another anther sample. For storage, prepare a plastic container with silica gel, a drying agent that prevents the deterioration of pollen caused by moisture. In order to do this, place the wooden container with the capsules on the silica gel. Otherwise, if such a container is not available, the capsules can be directly placed on the silica gel. Then, close and seal the plastic container with a piece of plastic sheet of extensible paraffin wax or parafilm wax, long enough so that it can be wrapped around several times to keep the container hermetically sealed. Finally, identify the plastic container and store it at 4 degrees Celsius, provided that the pollen is intended to be used within a maximum period of up to 2 weeks. Emasculation of the plants to be used as female progenitors. The flowers of the plants to be used as female progenitors are prepared for pollination during the early morning hours from 7 to 10 a.m. Begin this procedure by selecting an inflorescence that contains 5 to 6 flower buds of uniform size near their opening. Then, Remove all the open flowers and very small flower buds that have not been selected. Continue with the emasculation, the removal of the anthers of flower buds, to prevent self-fertilization. In order to achieve this, open each of the flower buds with tweezers or a stylus and remove the anthers carefully to avoid damaging the pistil. Thus, the flower buds will be ready for pollination the next day. Pollination. The next morning, the emasculated flower buds will open their petals fully, exposing the pistil, which will be receptive to pollination. Carefully open the gelatin capsule from the male progenitor you will use in pollination, according to the crossing plan. Do this close enough to the pistil to allow the stigma to make contact with the pollen. Repeat this procedure with each of the flowers of the inflorescence. Then, put the crosses label around the stem of the inflorescence. The label should identify the progenitors used, starting with the number or code of the female progenitor, followed by an X, and the number or code of the male progenitor, the number of flowers pollinated, and the date of pollination. At this point, the formation and development of berries will begin. During this period, attach nets to the peduncle that holds the berries in order to avoid losses due to their fall when they ripen. Harvesting and processing of berries. Maceration of berries and obtaining botanical seeds. Approximately 48 days following pollination, if the berries are fully developed, harvest them in kraft paper bags, duly identified with the name or the code of the progenitors that participated in the process, and the date and the number of the berries harvested. Then, leave the berries to mature inside the bag, in the environment, until they are soft and give off a particular odor, sign that they are ready to continue the process. Before beginning the seed extraction, place a parchment paper bag inside each kraft paper bag with the aforesaid identification information and the date of the extraction of the seeds. Following that, fill a pitcher with running water. One by one, squeeze out the berries immersed in water to release the seeds, process known as maceration. Upon completion of the maceration, remove some water and pass all the contents through a 25 mesh coarse net, which will prevent the remains of the carpels from passing through with the seeds. Thus, only the seeds will be received in another pitcher. Very carefully, rinse the seeds with water several times, making sure that they are not trapped in the remains of the net. Then, collect the seeds by pouring the contents of the latter pitcher in a thin piece of a 50 mesh net, previously folded in 4, forming a funnel to receive the seeds. Rinse the pitcher again with running water and pour it back into the net to retrieve all the seeds. Then, wash the seeds under running water to remove any traces of mucilage and rinse the net externally to completely clean it. Gather the seeds using a thin trowel. In this case, we're using a plastic stake, and place them in the corresponding parchment paper bag. Once closed, place the bags on a drying rack to allow them to dry at ambient temperature for approximately 6 hours. Cleaning, counting, dehumidifying, packaging, and final storage of the seeds. Once the seeds are dry, clean and count them in the laboratory. Previously, wash and disinfect your hands. Pour the seeds onto a piece of paper and, using a stylus previously disinfected with alcohol, separate the seeds that are blackened, empty, or without an embryo, while counting the visually healthy ones. If the seeds are abundant, an estimate of the total can be made with the help of a small test tube. Using a fine point indelible marker, mark the height reached by the volume of 200 healthy seeds. Then, using the test tube as the measure, calculate the amount of the selected seeds in the sample. After that, transfer the selected seeds, using a small funnel, to an aluminum foil envelope for its final drying. The envelope must display the code number of the progenitors, the year of crossing, the number of seeds, and the code assigned to the family, if any. Dry the seeds in a desiccator cabinet for as long as necessary, until the device registers a relative humidity of 4 to 5%, which can take from several days to several weeks. Otherwise, if a desiccator cabinet is not available, a container with silica gel in the bottom can be used. Upon completion of the drying time, remove the envelopes from the desiccator cabinet and seal them using a bag sealing machine. Afterwards, place the envelopes one by one in a larger aluminum foil envelope, which must display the same information written on the small envelope. If a barcode system is available, a self-sticking label containing the same information can be additionally placed. Finally, seal the envelopes with a bag sealing machine and transport them on a tray to their final storage destination in a cold room or refrigerator at 4 degrees Celsius. Seed viability test. Under the indicated storage conditions, the seeds will have a dormancy period of 6 to 24 months. Therefore, after 6 months, germination testing for the families projected for sowing can be initiated. For this purpose, place a sample of 100 seeds of each family in small plastic containers and take them to a humidity chamber for 24 hours. Then, take the samples out of the chamber and separate 2 replications of 50 seeds each. Place each one on filter paper into a properly identified petri dish of 9 centimeters. After that, moisten the filter paper by adding 5 milliliters of distilled water. Finally, incubate the petri dishes in a germination chamber or incubator at 27 degrees Celsius for 8 days. The emergence of the radicle is the criterion used to determine whether a seed has germinated. Results are expressed as a percentage of seeds germinated or viability percentage. Start the germinated seed count on the 3rd day, when the seeds begin to germinate, and perform it daily until the 8th day. Remove the germinated seeds to avoid confusion with the ones not germinated yet. Always keep the filter paper moist by adding distilled water when necessary. A germination percentage above 50 within 8 days indicates that the seeds of that family have broken dormancy and therefore are ready to be used immediately. Otherwise, place a sample in the incubator for additional 7 days at 17 degrees Celsius. If after this treatment the germination percentage is above 50, it is interpreted that the seeds of the family from which the sample was drawn were dormant. In case the sample of the seeds does not germinate after this second treatment, the seeds of that family are considered of poor quality and should be discarded. If you desire to accelerate the dormancy break of certain family seeds, you can store them from the beginning at 30 degrees Celsius at a moisture content below 5% for a period of 4 to 6 months. After this, take a sample for a germination test. If the seeds are still dormant, extend storage under the same conditions, testing for germination every 2 or 3 months until dormancy has been broken. Pollen viability assessment. Pollen viability assessment allows to identify infertile male progenitors within the target group. Pollen viability can be estimated by calculating the percentage of viable pollen by means of the dye test with acetocarmine glycerol jelly or the percentage of germinated pollen in the germination test in vitro. Acetocarmine glycerol jelly test. For the acetocarmine glycerol jelly test, place 1 or 2 drops of 2% acetocarmine glycerol jelly in the center of a slide, previously identified with the progenitor's code. Following that, using a wooden toothpick, take a small amount of pollen out of the capsule and place it on the drop of dye, spreading the pollen with light circular movements. Let it stand for a minute and then put the slide cover on. Mounted slides should lie flat for 1 or 2 days. Afterwards, observe them under a light microscope at 400x magnification. A bright red staining of the cytoplasm of a pollen grain is indicative of viable pollen, while cytoplasm not red or pink indicates non-viable pollen. We recommend the evaluation of 250 pollen grains, expressing the results as a percentage of the viable pollen. A progenitor is considered fertile if more than 50% of the pollen sample is viable. The observation of abnormalities such as tetrads or pollen grains with 4 cores indicates that the pollen sample is infertile. In samples of pollen from diploid potatoes, pollen grains may be larger than normal grains, in which case they correspond to pollen with a non-reduced genetic load. This phenomenon of unreduced gametes is rarely observed in tetraploid potatoes. In vitro pollen germination. In vitro pollen germination is a test that simulates the pollinic tube growth in the cellular tissue, given that the culture medium used in its composition resembles the stigma mucilate. In a beaker of 100 milliliters, add 5 milliliters of a solution of boric acid, 20 grams of sucrose, and 0.2 milliliters of polysorbate 20, commercially known as Twin 20. Complete with 100 milliliters of distilled water and take to a magnetic stirrer to homogenize the solution, which should be at pH 5.5. Once the solution is ready, place 4 drops on the base of a 5.5 centimeter petri dish, distributing them in the vertices of an imaginary square. Then, with the tip of a wooden toothpick, add small amounts of pollen from the same sample on each drop, spreading the pollen with light circular movements. Separately, use the lid of the petri dish to prepare a humidity chamber by placing a filter paper moistened with distilled water covering the bottom of the lid. Cover the bases of the petri dishes with the lids and leave them overnight at a temperature of 20 to 24 degrees Celsius. The next day, place a drop of iodine solution on the sample previously laid on the base of the petri dish and cover it with a 22 by 40 millimeter slide. After that, observe it under a light microscope with 100x magnification. A pollen grain is considered to have successfully germinated when the pollinic tube length reaches a size equal to or greater than the diameter of the pollen grain. Germinated and ungerminated pollen grains count must be performed at a minimum of 10 fields. Results are expressed as the percentage of the total number of pollen grains per field. An average rate of 80% indicates fertile pollen.